Thanks a lot, Nicole, John, for hosting the session, and Thomas Lehner for making this all happen. Sue Grapper, who was supposed to give this presentation, her vocal cords were victim of the festivities last night, so she can't really speak. I'm a last-minute replacement here. It was a great party. I'd like to kind of build this bridge between phenotypic screening and machine learning as an enhanced readout to make our phenotypic assays more relevant than just relying on standard biochemical readouts. We are interested in NASH, non-alcoholic steatohepatitis. Why is that so interesting? Well, first of all, it's a quite complex disease that needs lipid uptake in the liver, inflammation, and then fibrosis, cirrhosis. Secondly, there is no current therapeutic solution on the market. And I think it's a great example how 3D microtissues can be used to model a complex disease in vitro, and how we can read these systems out. So the three aspects that I'm going to touch today is how are we building this model and how are we stimulating the model to develop the disease phenotype? What are the endpoints and readouts that we're using to measure what's going on? And then how do we use image-based or machine learning-based image quantification to measure the complex morphology of the fibrils that are formed during that fibrotic step? So the system is based on our gold standard liver tox model, which is a co-culture model of hepatocytes and non-parenchymal cells like Kupffer cells and endothelial cells. But what we add to the system are stellate cells, which are extremely important to develop the fibrotic disease phenotype that we are after. Now, we start with a lean and healthy model, and then we induce NASH by feeding the tissue a cocktail of free fatty acids and high glucose concentration. So it's basically a diabetic medium. It takes a few days until the tissues take up the lipids and then start to develop a disease phenotype. But we also have to create an inflammatory insult using bacterial LPS that then triggers the disease progression in the system. And now that we have a diseased phenotype, we can start to look at compounds that are either anti-inflammatory, anti-fibrotic, or metabolic to reduce lipid uptake in such a system. What's important is that we have a pretty substantial battery of assay endpoints that, on the one hand, are screening compatible because we want to upscale that system for automated screening. On the other hand, we also have endpoints that have high clinical relevance because ultimately we want to convince pathologists and researchers that this is a functional model and that they can relate this also to clinical endpoints. So the primary ones that you see today are biochemical assays on the screening side, and then either high content imaging based or histology based IHC endpoints to look at tissue morphology from a histological perspective. So this happens all in our Acura 96 well plates, a single tissue per well, always the same cellular composition, always the same size. Uniformity is key here because otherwise we'll never really get to a screening compatible solution. So let me take you through the three hallmarks of NASH: steatosis, inflammation, and fibrosis. And we start with steatosis. What you see on the left are high content images of these tissues. We use Nile red staining to make the lipid uptake visible. You see DAPI for the nuclei in the system. The lean control is on the left, and then the induced NASH is on the right. And you see a clear increase in Nile red staining here. If we just by pixel counting do a simple quantification of this image, we already see that we have a 10x increase from control to NASH condition. So it's a very strong signal. We have a great dynamic window in this model. The imaging endpoint is great, but it's fairly time consuming to generate this data. So for a screening application, we'd rather prefer to have a biochemical readout, and that's what you see on the right side. Using a triglyceride assay, we can pretty much generate the same data. We see an 8 times increase here between control and NASH. So again, very large dynamic assay window in this system. Now for the inflammatory response, we use a Luminex assay. and we're able to quantify a large number of cytokines and chemokines in parallel with the system. We have built panels that help us to look at pro-inflammatory markers like IL-6 and IL-1 beta, or at fibrotic markers like TNF-alpha and IL-10 in parallel in such a system. And what you see is that between control and the NASH model, we see a substantial increase in chemokines, in cytokines across the board. And this has also been true for chemokines on this slide, where we again have a good mix of pro-fibrotic and pro-inflammatory markers in the system. Now, the important aspect is how are we quantifying fibrosis in such a context? Because it's not just about the amount of collagen that's deposited in the system. It's also about the structure of the fibrils, how mature they are, how strongly assembled they are. And I'll show you how we attack that in this proof of concept study. We took an antibody, an anti-TGF beta-1 antibody. Why is this relevant in this context? TGF beta-1 is one of the drivers of fibrosis in endothelial cells. So you see that the TGF beta-1 triggers the TGF receptor 2, which then the receptor 1 recruits ALK1, ALK5, and then triggers this whole signaling chain through SMAT, and ultimately leads to an upregulation of collagens and TIMS. And if we are blocking the TGF beta-1, we should see a decrease in collagens and TIMS in the system. And that's what I'm going to show you in the next couple of slides. So our screening compatible endpoint to look at collagen is measuring procollagen 1 and 3 in the supernatant. You see the procollagen 3 data on this slide. What's really nice is you see the dose response that works both on day 5 and on day 7 to 10 in this data set. We have a pretty good consistency from model to model, so high statistical relevance in this assay. And what's important to note here, this is a measurement that's taken in supernatant, so we can also look at longitudinal changes over time inside the same tissue and multiplex this with inflammatory marker quantification using the Luminix assay. So you get a lot of data from this complex model over time. On the next slide, you see the same quantification now for tissue inhibitor of metalloproteases, TIM. The TIMs are very important in NASH. Typically in NASH patients, the TIMs are upregulated. The TIMs then inhibit the MMPs in the patient, and the MMPs are necessary to break down extracellular matrix molecules. So if TIMs are high, then MMPs cannot work, and the extra collagen that's deposited in a NASH liver cannot be removed. And so we want to change that by a pharmaceutical intervention. And you see that as we add the anti-TGF beta-1 antibody, we see a substantial reduction in TIM1 and TIM2. So again, correlation to the clinical findings for this antibody. Now, the TIMs and procollagen have another important significance for us because they are the two of the 3 aspects of the so-called ELF diagnostic test. The ELF test has been developed by Gilead and Siemens. It's one of the few non-invasive diagnostic tests for NASH. The third quantity in that assay is hyaluronic acid. So PC3, TIMP1, and hyaluronic acid together are part of this ELF test. And this is kind of where we go full circle because we want to have readouts in the in vitro case that have a clear relevance to the clinical setting. Now, what are we doing about fibrosis? We are able to look at procollagen in the supernatant. It's a precursor of the collagen deposition. But of course, we really want to have a good understanding on a morphological level what happens in these microtissues so that we can assess the impact of a drug on fibril deposition, and also the formation of fibrotic fiber networks in these systems. We've started about 1.5 years ago to collaborate with a company in the US called PharmaNest, and they've developed an artificial intelligence or machine learning based approach to quantify fibril networks on sections to allow us to get a better understanding of how a drug intervention changes the morphology of the fibers that are deposited in the system. And this is quite complex. You see the serous red staining for fibers on this image on the left side. If we're again just doing a pixel quantification, we're losing a lot of biological information that's actually contained in these models. So the quantification approach in the FibroNest system consists of 3 different buckets. The first bucket is how much fibrils do we have and what is their overall structure. The second aspect is what's their shape. That's the morphometric approach. And then the third aspect is where are the fibers across this tissue and how are they organized? Are they fully assembled? How many nodes do they have typically per fiber and so on. So using that approach, we get about 350 to 400 unique quantifiable fibrosis parameters. And based on these parameters, we've developed a compound score that takes basically a weighted sum of 37 of these parameters to create a compounded index that gives us a measure of fibrosis that we have in the system. So how does that work practically? You see the serous red bright field image on the top with already a visible change or visible reduction of fibril deposition as we add our TGF-beta1 antibody to the system. But the FibroNest software now allows us to really understand what kind of morphological changes we see. So in the second line, you see in pink the assembled collagen and in green the fine collagen. The fine collagen fibers are fairly young collagen fibers that are not yet fully embedded into assembled networks. They are an important drug target for us because they are much easier decomposed by MMPs in the system than a much more solid and rigid assembled fibril network. And then on the lower end, you see dense and faint collagen. So we get a lot of extra information from this machine learning approach. But of course, we want to ultimately have a kind of understanding of what happens to these tissues. And that's where a heat map is really quite helpful. So you see the control NASH and then 2 concentrations of the TGF-beta antibody. Each column that you see here is a different region of interest, and the region of interest for us is one tissue in the section. We are typically embedding 12 to 16 tissues into one block before we section it so that we have a bit more histological real estate to generate data from. And the algorithm is automatically identifying the tissues and then numbering them as regions of interest. And so what we get are very interesting fingerprints that show us how specific drugs change these sets of parameters in their specific way. So you see the control on the left side. In NASH, all of the parameters basically get red. And then we see a concentration dependence as we add the anti-TGF-beta antibody to the system. So a big question here is what is the physiological meaning of these different parameters? How does it actually link to clinical aspects and how does it link to patient outcomes? And I'll come to that in my last slide because this is a subject of current investigation with our partners. The main focus on how we use this approach at the moment is that we are using different compounded indices that are basically drafting information from a selected subset of the 400 quantifiable fibrosis parameters and then give us a compounded score that is a figure of merit for us to decide, is this a drug that has an antifibrotic effect or not? And is that effect dose dependent, which are two key criteria for moving forward with the drug in the clinical trials. Now, where do we want to take this technology, and why are we so interested in scaling it up, both on the biological side, by being able to have miniaturized uniform tissues in a 384-well format, but also on the assay side to be able to generate data at fairly low cost. The reason is that we believe that a key to solving the NASH problem in drug discovery is to use combination drug combinations to address the different mechanistic effects that lead to the disease. NASH is an extremely complex disease that involves dietary aspects, cardiovascular aspect, metabolic aspects, gene predisposition. So to be able to attack such a complex disease, most likely we need more than just a single compound, but a combination of compounds. And this makes the whole drug discovery aspect a lot more complex, and it requires a lot more data. And that this is actually helpful, we've shown in a study together with Gilead. You see the results here at the lower end here. What we did was looking at elafibranor and firsocostat, both drugs that didn't really make it through the clinic. You see that each of the drugs, this is control, this is NASH, each of the drugs in itself is not very powerful, but when we start combining the two drugs, we see a synergistic effects by this drug-drug combination. And this, for us, is a way to go, also with other drugs under investigation, to see how we can improve the efficacy by combining them in the right combination. So to be able to do that, we need a high throughput compatible solution. We need to be able to measure the hallmarks of NASH in a highly integrated, low-cost biochemical assay setting, but we also need to be able to verify our findings in clinical relevant readouts. And all of these aspects we can fulfill with a fairly simple and easy to scale up spheroid system. As I mentioned, one of the aspects of investigation is how can we relate our in vitro findings to actual patient outcomes. And we have a poster about this at the EASL conference in 2 weeks, collaboration with Fibronest and Arun Sanyal at Virginia Commonwealth Hospital, where we are taking data from 19 NASH biopsies from actual patients and then benchmarking them against our in vitro findings. And what I can already say is that 90 out of these 400 fibrotic parameters show an excellent correlation with clinical outcomes. So we're extremely happy about this. Let me thank the liver disease group that at Inspiro that made all this work possible. Francisco Verdeguer is our VP working on this with his team, and also Mathieu Petitjean and his colleagues at PharmaNest here in the US for helping us with the machine learning. And thanks very much for your attention.
We have time for a question or two, and then there will be a brief discussion of the session. One of the great advantages of organoids is that you have lots of them. So if you look at the multi-drug combinatoric problem, have you done a power analysis to figure out how many organoids you would have to do to design a multi-drug cocktail?
Yeah, that's a great question. So we haven't looked at it from this angle. The way how we want to approach the combination testing is actually on a more iterative level. So by selecting compound sets with different modes of actions to see which type of compound in combination is moving the needle, and then improving these compound sets iteratively as we go to zoom in on a combination that works.
There are questions, though, about if you have nonlinear interactions, that becomes a little bit more complicated on the iteration.
Agreed with that, and we've seen that in a different context in combination treatments where the response was nonlinear in the sense that sometimes lower concentrations were actually showing higher efficacy than higher concentrations of combination drugs. So there is a risk with this approach that we are kind of falling into kind of a local maximum, but we're not really getting the best combination. But I think that's a bit of a trade-off between the size of the screen and the capabilities that we have.
Okay, thank you very much. Oh, we have a question. Good, great.
My name is Job Bomen from the University of Twente. Thanks for the very interesting talk, and especially comparing it to the pathology in patients. What I was wondering, do you consider having a test and a train set with the patient samples? Because you're now going from 100 parameters in all potential combinations and permutations.
Yeah, definitely. So I think in the current setting, from the 90 biopsies, and this is not a large number, I'm totally aware, right? We've selected 15 for a blinded validation test. Or qualification test, I should rather say, and the others are used for training the system.
And these biopsies, how do they relate to the clinical outcome?
So the biopsies are primarily at the moment relating to the stage of NASH that the patient has, right? So as we don't really have a pharmaceutical intervention against NASH, it's hard to, we don't have responders and non-responders, right? But we have different progression of the disease. And our main interest at the moment is that we are able to basically classify the 3D tissues into the same buckets as a human pathologist would classify the biopsies. That's limited to a certain extent because clearly the tissues are quite small. They lack some of the features of a biopsy. You don't have a portal vein, for instance. So I think there is still a bit of translation that we need to do. But on the other hand, a big benefit is that our calculated index is continuous. So we're not just looking at like 5 classifications, 1 to 5, but we can also have a much more granular assessment. And I think this will be crucial to see these fine changes that some of the pharmaceutical interventions will entail.
Thank you.
I can't imagine that Lance has anything to say about the liver, but I would love to hear his question. Lance?
Jan, very nice talk.
Thank you.
Are you able to control the oxygen tension in your plates? And what is the oxygen tension? And does it relate more to zone 1 or zone 3? Because there are differences.
That's a great question. So we don't see zonation in these tissues. It's also not really expected due to the size of the tissues. But it's been a recurring topic for us. So I think one of the interests that we have on the microfluidics aspects of microphysiological systems is actually at some point creating a liver system that would allow us to show zonation in that device. And if you have a good idea, Lance, then I'm happy to pick it up.
Use the mic.
Lance, can you use the microphone?
So Lance's point was uniformity and how you could drive a single tissue into a specific zone. So that's something that we're currently not doing. We're keeping constant oxygen tension. Yeah.
Well, one of the interesting questions following up on Lance's comment or request for information about whether you're doing oxygen tension is whether, in fact, zonation can be studied with independent zones. So you build a plate that's oxygenated and a plate that isn't, or has a lower oxygen and even lower for, let's say, the third zone, and the extent to which they have to talk to each other or can be treated independently. I would like to hear from either you or Lance about the liver physiology on things like zonation.
Yeah. So I think the two possibilities to address this is, as you said, it could be a discrete system where we have different types of tissues that are cultured under different conditions, but you could also imagine a tissue culture that has an imposed oxygen gradient, so that you can drive the system as in a holistic manner in this field. I think our primary interest here is building a system that allows us to run a screen to find compounds against the disease in an efficient way. So we've decided not to add these extra layers of complexity to the system. But from a scientific perspective, I totally see the point. Yeah.
But in the Inspiro plates, you have serial perfusion of organoids. And the net result is that there can be metabolic gradients along that flow, depending upon your flow rate. Because Kim and Ingber, looking at the CACO2 cells, clearly showed that there was a difference in expression, or this happened to be of villi, but it is a general phenomenon along the length of a channel. So to some extent, you have a situation with spheroid after spheroid after spheroid, where there's going to be, by definition, a metabolic gradient, depending upon your flow rate.
Yeah. So the work that we do here is under static conditions in the 384 well system, right? So we have no flow because again, this is a screening setup, but point is well taken. We also have the Acura flow system where we are lining up tissues. Olivier and his team and our colleagues at ETH have done quite a lot of computational fluidics analysis. So it really depends on the cell size versus medium volume that you would really see a depletion of specific nutrients or an increase of, let's say, metabolic byproducts, right? But this definitely plays a role. Yeah.